My name is Olivia Collins. I am a physician assistant major at Gannon University in Erie, Pennsylvania. My passion is genetic research. Over the past 2 summers, I worked at the Cleveland Clinic's Cytogenetics and Molecular Pathology Lab. In this clinical setting, I found my passion for research and the desire to incorporate medical research into my future career. For my first project, I focused on using a combination of purified interleukins 4 and 6 to stimulate multiple myeloma cell growth. Interleukins are a type of cytokine that stimulates cell proliferation. The goal of this study was to produce enough cell growth to be able to increase the detection of chromosomal abnormalities in white blood cells. In the end, I realized that the 2 interleukins used counteracted each other and did not result in ideal cell growth or the expected abnormalities. If the interleukins had been able to work together, the genetic information gathered from the cells would indicate specific translocations that signify the severity of a patient's multiple myeloma. With this information, clinicians would be able to create a treatment plan specific to the severity of each patient's condition. When I returned the following summer, I studied white blood cells from blood samples and the effects of drug-induced early mitosis, or premature chromosome condensation. Abbreviated PCC, premature chromosome condensation, is a technique used by radiation biology and chromosome science to stimulate mitosis during cell interphase. In order to introduce early mitosis, chemicals that inhibit protein phosphatase activity were used. Protein phosphatases are responsible for controlling interphase and cell transformation. When acted on by oxalic acid or colliculin A, the DNA inside the cell nuclei rapidly condenses into small chromosomes. In the comparison study, colliculin A was more effective than oxalic acid in the control groups. The experiment results can be applied to patient cells that cannot reach normal cell division, such as those exposed to radiation. Radiation exposure inhibits the standard metaphase chromosome analysis. By using colliculin A, a condensed chromosome analysis would be able to indicate the amount of radiation that a patient was exposed to. The higher the number of genetic abnormalities, the greater the amount of radiation the patient was exposed to. In the future, I hope to introduce medical research into the growing roles of physician assistants. I believe that there is a need for a stronger relationship between physicians and researchers to make the healthcare team more connected. Thank you for your consideration.